Hi, everybody, and welcome to Neuroscientists Talk Shop, UTSA's Neuroscience Research Podcast. Today is February 5th, 2026, and we have Dr. Jonathan Bohmann with us. Jonathan is a staff scientist at Southwest Research Institute here in San Antonio, and he leads a group of scientists doing medicinal and computational chemistry. He's an expert on the relationships between molecular structure and biological activity of receptors and enzymes and perhaps other large molecules that I don't know about, and the complex set of experimental and analytical techniques that are used to discover how those molecules actually work as little machines that do work and give off or absorb energy. And we need to know that to understand how they work, partly just because there are properties of the molecules that are surprising and that we need to know about, but also partly because we want to make drugs that interact with the molecule interfere or enhance the function of the molecule. And so that's the medicinal chemistry sort of side of all of that. So hi, Jonathan.
Hi.
Welcome. So here today with us is also Hsieh. Jenny is chair of the Department of Neuroscience, Developmental, and Regenerative Biology at UTSA, and a podcast favorite. Hi, Jenny.
Hello.
And I'm Charlie Wilson. So, Jonathan, acetylcholine is kind of unusual among the small molecule neurotransmitters because it has a ubiquitous enzyme in the extracellular space that degrades it, and that's how it gets removed from the synaptic cleft. And most of the other small molecule transmitters diffuse away and get absorbed by reuptake. And that is a cool property of that kind of synapse and depends very much on that enzyme to sweep up all the acetylcholine that was released. It even is a barrier, and the acetylcholine has to penetrate that barrier somehow to even get to the postsynaptic membrane. And if it bounces off the postsynaptic membrane, it gets eaten up then, doesn't get a chance to do it again.
Right.
And so it's a really great way to make very, very precisely timed synaptic effects. And it's a puzzle how that molecule works. It works really well and really fast. And so one of the questions we'd like to understand is, how does it do that? And just as from my naive point of view, not normally working on this, I'm picturing the enzyme as a little machine, and it's got moving parts. And then acetylcholine comes in there and interacts with some of those parts, and the others move. The acetylcholine gets broken apart. Then somehow the degradation products have to be spit out really quickly, and then has to get ready to do it again. And you sort of like to imagine that. So that's one reason, in my mind, of wanting to understand this molecular structure thing. Maybe a more practical reason is you want to mess with the molecule somehow. You want to inhibit it or enhance it, or maybe something's made it go wrong and you'd like to fix it.
Right.
So both of those are reasons for us to want to understand this stuff. So, and acetylcholinesterase is one of the proteins that's been studied this way for a long time. And so maybe we know quite a bit about it, and hopefully you can fill us in on some of it. But I'd really like to ask, sort of, how did we get here? How do we learn this stuff? What are the raw data that we have to use? What are the challenges that data presents to us as we try to turn them into a story about how the molecule works?
Well, of course, depends on how far we want to go back. But
Not too far back.
Of course, the foundational understanding begin with the postulates about the active site. Without any other information, we know that the complementary features to the neurotransmitter are needed, and so you had identified early on an anionic site or a ester pocket, surrounding that catalytic. They were identified based on the substrate functionality or complements to it. So then what the surprising part was when the structures were resolved, which is how deep the channel from the periphery to the active residues actually is, and it's something like 14 angstroms or more. So it's a real puzzle because the turnover rate, the just incredible turnover rate of the enzyme would, to support that, you'd have to imagine somehow that the products of the enzymatic reaction would exit and somehow have to pass the incoming substrate.
Yeah,
and that seems it's hard work to get in and it's hard work to get out. And so the implication is that there must be another exit. And this has been the mysterious, what they call the back door effect. And what the interesting thing would be, what if there was a doorman to help out with that? And we've had questions about that, but the starting point is the structural information that's available. The interesting part is that there's a loop there called the omega loop, which really is at that junction where we could imagine the thinnest part of that wall, the catalytic pocket wall being, and that's where this presumed exit would be. But over the course of decades, as structural biologists have purified and crystallized proteins with every kind of inhibitor that's been isolated, and every kind of species from which the enzyme has been expressed, there's really not evidence of, there's really tight alignment of those structures at the site of that omega loop, when there really is more flexibility in other regions of the enzyme. It's obvious that an enzyme to function like that has to have some.
So one of these crystal structures is a snapshot of the molecule,
right?
And then the idea is you put a inhibitor or a substrate or something in there, and then crystallize it with that in place,
right.
And you get another still shot,
right?
But now with the thing in place,
right?
So that's telling you where it sticks. But what it doesn't tell you is how anything gets.
That's right, because the gorge, the opening in those states would not permit the entry of the molecule that's been co-crystallized in there.
So people probably hoped that with one of these in there, they'd get a picture with the back door open.
Right.
And that just hasn't happened.
It has not happened at all.
It could happen still.
Right.
But we beyond just the structure, you also have to know about the chemistry of the interaction between the substrate and the protein.
Yeah.
So you have to be able to look at it and say, this kind of residue would interact with this part of the acetylcholine molecule. And that's sort of regular chemistry.
Right.
And is that like super well known? I could just look at the structure and say, oh, this part would bind an acetylcholine for sure.
Right. Well, actually, in many of it's retrospectively interesting to look at known inhibitors, and you could say, all right, what I need is, it's a pharmacophore. It's a set of essential features, and I need maybe a cationic nitrogen center, and I need some sort of polar ester-like things, a model. I mean, all of the inhibitors that have been intentionally designed, in history, I guess, the historical the legacy, were all designed or connected with acetylcholine with those features in mind. So it's not too much of a, we can retrospectively look at molecules that are inhibitors and reason why they would be inhibitors to begin with, but that doesn't necessarily provide a insight, prospective, because
what you want.
that's right. because what we know is that pocket now over time, the more data that we get, the more diverse we're seeing that enzyme can be in terms of the arrangement near the active site in these other pockets. So you can look at now we found yet another inhibitor or substrate like molecule. Oh, well, let's adjust. This bigger thing now actually will fit. So maybe our reasoning wasn't quite what we thought. So we're learning that the regions in the acyl pocket, which is adjacent to the active site, are quite flexible. If we look in my experience in the modeling that I've done on the enzyme, there's really an opening that consists of 3 tyrosines. In some species, it's a phenylalanine that these, in addition to that acyl pocket, can open or close, well, much like one of the doors on Star Trek. It looks like these, and there are definitely structures where those phenylalanines, or in the case of most, the tyrosine residues, can literally flip open and closed to either block or open the active site.
And you know that because you've got structures in which they were open and structures in which
when they're closed. Yeah.
So it's not because you have you can from first principles say, oh, this piece of the molecule will be able to open and close.
We rely on the structural information, that the evidence that's there, at least for these, frozen structures. And we can say, and the interesting thing too is what we're learning, it's kind of unexpected. Some of the work that I did earlier in my career was trying to develop models that were more specific to what we would expect for human cholinesterase versus murine. And so the murine structures in that study were rejected or not included because we wanted to focus on, well, it turns out we really needed the information that just happened to have been captured in the mouse structures.
So are you saying that the crystal structures could potentially be different based on the species?
Yes, they can be. Yes.
And how does one decide which species to
use as a model?
Yes.
Oh, well, that's we haven't done it in the lab that I run. I think what we're interested in is keeping our mind open, and we try to make these into well-defined computer experiments. So the key is a workflow that when we say, all right, here's a crystal structure we haven't used before, then how would that change the predictions that we've made? And so what we want to do is, the key to making that all work, including a new crystal structure deciding, is how does that impact the outcome of my workflow? So I set up a test bed, a model of what a computational workflow could be like. And then I say, as the area of fundamental research, what is the effect of the choice of my crystal structure on the outcome of that workflow? So the point isn't the discovery at that point. The point is the workflow in the discovery. And that's where my passion is in designing the workflow, not necessarily engineering the discovery itself, because that's something that people that work in my lab will do. But my interest is exactly the question. We can't model or flexibility with a static crystal structure, but we can certainly ask the question, at the end of the day, what's the impact of using a murine crystal structure on a human to make predictions about what will happen in an assay that is based on a human expressed protein. Should we be doing that? Does it make sense? Well, the way that our work, what we do, is set up. Did I say that? Yeah.
Yeah.
So our focus is on what is the impact of those choices on the computational outcome? Not what is the outcome, but how do things like the choice of a murine versus a human, the crystal, what happened with the murine versus human is it wasn't even a good comparison because they crystallized in different point groups. So you had that problem too. It wasn't
What's a
just
point group?
it was the symmetry group in the crystal that was used to refine the crystal structure.
How you go from the image into the
right,
structure.
exactly. Yes. So you had not only the species differences, but then superimposed on that, they were actually crystallized in different forms. They were not the same crystalline form. So how do you unwind all that? Well, in one of our projects, we found out that using a murine crystal structure gave us an insight into how a portion of the pocket could be leveraged for one of our drug discovery projects. That ended up having an impact, even though this was targeted for a human enzyme. So I think in the case, at least, of cholinesterase, what these different species, when we found out it wasn't right to say we have a focus on human therapeutics, we'll only use the human crystal structures. That was not right. And we had a lot of benefit in our workflow, at least this is just from a computational workflow of including that other structural information, including the residue flexibility that we found in a murine crystal structure.
So let me try to understand what the model consists of. So the model is a collection of knowledge that you have about how molecules interact with each other.
Yeah.
And then you give it this structure, and then it will say, and if you tried to put an acetylcholine here, it wouldn't fit.
Right.
Or it wouldn't bind.
Right.
And then you could ask those kinds of questions about the whole structure. That's basically
that's basically it. Right. So the foundation is the laboratory data, which is the three-dimensional crystal structure. But on top of that, then we have an algorithm that will take the atomic coordinates and translate that into a penalty function, for example, or an optimization function if you'd like to go from positive to negative, but then that creates some sort of surface. And so the core of it is there's the crystal structure that we use, but there's also choices about other parts of the model, if you will, the coefficients, how the model is trained to make predictions that matter too.
It includes energetics, like
it's
an energetically favorable.
Right. Yeah, mostly enthalpy-based models. So we need to pre-screen our projects if we can to decide whether, it's largely enthalpy controlled or entropy controlled. We prefer the enthalpy controlled situations. Those make better models.
Because they have fewer moving parts. Is that the reason?
There's a better connection between enthalpic contributions to binding and the structure than there are the entropic components, because entropy is about structuring penalties, and it's hard to see what those penalties are from a solid state representation.
Oh yeah. If you could see it, if you could actually have a movie, then that would tell you.
Well, people have done that, and they've found, ways that we've talked about the back door, ways that could open in those dynamic situations. But getting back to the models, in other words, so we did an experiment a few years ago to look at basically 50 different workflows to arrive at a activity prediction. So those workflows would involve choosing between 5 different crystal structures, choosing different strategies for pre-processing a data set that might have active compounds versus inactive compounds in order to train the model, right? So one of the chief problems with machine learning and drug discovery is that the data is very imbalanced and biased because people only put out the best of their laboratory results. They don't include the worst of the worst. So those data sets are unbalanced. So the interesting thing is we looked at the different crystal structures and then different ways of balancing the data sets. That was the second thing. And then now you have the options of different ways of combining that data. So those are your actual machine architectures. We use something called support vector machines. We can use neural networks, et cetera. There's a variety of those. So in this study, we used 3 of those. And you can, but we structured this. And the important thing was, when you looked at the model performance in predicting new cholinesterase inhibitors, there's only one thing that drove the performance, and that is the choice of crystal structure that you use. So everything hinges on
what I'd hoped.
Yeah.
I'd hoped that they would all, the important part would be conserved. And,
Right. No. Yeah. And so the only thing that matters, and you've seen this in, there was several years ago a contest at University of California, San Diego, the pharmacology, Skaggs School of Pharmacy, Michael Gilson hosted something called, the Grand Challenge contest, and there's others like it. But one of the conclusions was, after assembling, it was between 50 and 100 different research groups and having them submit predictions in a similar fashion, that you could have research groups using the same computational tools come out with different predictions. And the difference was details in the workflow. So just like in the laboratory, in computational predictions, workflow is everything. It's not a guarantee to get a useful result with just a workflow that you decide ahead of. There needs to be research on what are the best practices. And then the nice thing is, with the processing power that's available, you can make a computer experiment with a baseline and control, just like you would in the laboratory. And I think the most important progress we've made, or the most successful projects, have been where we've had a strong baseline. We always want that, but then where we've had the resources to do complete test of different workflows. So just the prediction isn't the interesting part. The interesting thing is the impact of different workflows on that prediction.
So you could train a machine
machine to test all the workflows.
If you had enough data.
Right. Well, that could be it. But then, I think where people are interested in the machine learning with that is not so much, that is the question that would be the most impactful, right? And I think there's convergence, and I think the way that what's probably good about one of the things that's very promising about the emergence of some of the latest larger machine learning models, and especially the agents, is why not have an agent. We don't have to get necessarily smarter about the simulations, but let's program agents to run simulations with different starting conditions, and then merging the data at the end, right? So we know how changes in the modeling workflow when it comes to cholinesterase or any of the other targets have an impact. But we were talking about different crystal structures, and it is good news that those every one of those different crystal structures encodes information that's impactful. And so I think that's
that's also rationale why motivating better crystal structures.
It is. But it's an interesting thing too. I'm a proponent of taking all available information. And that does include, now the interesting thing is what happens with obviously machine-generated biostructures.
Like
But you look at,
AlphaFold?
yeah, but you look at an interesting thing with cholinesterase is that the majority, if not the entirety, of cholinesterase crystal structures doesn't really give a strong mechanistic picture of just the processing of the natural substrate, right? That omega loop is frozen. But now those 200 or 300 crystal structures are now what AlphaFold knows about cholinesterase.
So there are little differences between them that would give AlphaFold some information about the degrees of freedom of movement in there.
Well, we'll see what happens. I don't know. That's on my to-do list. But one outcome is that AlphaFold just simply is so strongly trained by all those examples where there's no flexibility that it would produce a model where flexibility is not going to be captured. Or there could be a more creative way to include structural information from enzymes that maybe share some common structural motifs. And then the interesting thing would be to now say, all right, given the appearance of these cholinesterase models, plus some partial alignment on these related enzymes that are known to have a thinner wall or an opening, what would be the projection of these, diverse, the sequence of acetylcholine on these backbone structures that permit an opening? And I think that'd be a specific machine problem that would be a really interesting one.
So one of the reasons the reason we end up with still photographs of this protein structure is because the cameras that we use require that they sit still.
Right, yes.
It's just like regular cameras used to require that people sit still, and you got these pictures of people holding their breath. And what are the prospects of a better way of imaging the molecular structure that would allow it to be more flexible?
Of course, for high enough molecular weight proteins, there's cryo-EM. But the interesting thing from the drug discovery side is that as I mentioned from the experiments that we've done with the impact of crystal structure choice on the prediction, is it's small changes near the binding site, like the displacement of a single residue.
It might be very tiny.
That would be small, that are still impactful, are encoded in the machine learning, and have an impact on the outcome of the prediction. That's another seminar. But I guess where I was going with that is if those small structural, differences have an impact on the modeling, then I think you'd have to focus on, in some day, if you could get a cryo-EM, so to speak, on something like cholinesterase, it also would have to make it comparable, you'd have to have a refinement,
you need super high resolution.
A higher resolution
short
enough,
exposure.
which we don't have that right now, obviously, with cryo-EM.
And of course, even cryo-EM, it's frozen. That's what cryo means.
That's what it means, right?
It's not that.
But I think there's an opportunity there, at least for when you talk about the effects of crystal packing forces, then that is a way to at least start to separate that out. What's the influence of crystal packing versus just low temperature, right?
So maybe I shift gears a little bit and kind of ask you the reason why you were interested in developing a pharmacophore model around acetylcholinesterase is because there are development of molecules that would inhibit, and these are so-called chemical weapons. And you were interested in reactivating and this is where the science started.
Right,
So
right.
how do we know that there are bad players who would then not want to extend your work and find inhibitors of your reactivators?
Oh, yeah.
And where does it keep?
Where does it stop?
Sort of this battle.
I'm not sure what the answer is with that. I really don't know. But I think the issue.
And can AI be now?
Yeah, I
used?
think the bigger issue. I think there was a demonstration of this a few years ago. I mean, that's the way that AI can be used. That's not necessarily the way that we want to go. Is that people just design things that are nasty?
Yeah.
And that, it's interesting. I tried to. I'm not worried about the large language models today, but it doesn't take too much imagination to say, all right, large language model, you've learned enough chemistry, now I have this thing. And I don't know what the controls are on large language models today about designing or anti-designing things. But I think this was, I'm getting off the page here. The concern about this kind of thing has been raised and that was a Nature paper a few years ago. I think some of us have seen it.
Mm-hmm.
And it's definitely on my mind, and other people have that on their mind too. I don't know what to say about it, but we know that it's out there. Yeah.
Cholinesterase inhibitors are one of the oldest
They are one of the
chemical
oldest
warfare agents.
Yeah, it's interesting that the story is and I could be fact checked on all this. But originally the intent was insect control. But Gerhard Schrader of, I think, BASF, this is public, I don't want to get it wrong, but back in the 30s, then developed chemical weapons, and that goes back to Fritz Haber and Lewis, and then Lewis developed Lewisite, that was the arsenic. And then kind of this all happened at the same time, but World War I and then World War II saw the emergence of sarin and some of the other taboon all came out of the Schrader work. Of course, those were designed, initially that work came out of an agricultural industry, but then it was discovered that they were very highly toxic. So that's one of the things that doesn't carry over from species to species, is we know that from the progress that's been made, or the sequence of events that happened with the organophosphate compounds, whether in the application to insecticides or others, that there's a very exquisite species selectivity, and ends up being for those inhibitors. So they're very, the insect, I guess, toxins have been very designed to be very selective for that cholinesterase, and some of those are, like peroxon, or 10,000. I'm just making up a number here, but several thousand times less toxic than some of the other agents are. So this is an interesting thing, is that we're able to leverage some of those other crystal structures in our work, but we know that fundamentally their receptivity to these different organophosphates is a lot different. I'm not saying but it's broadly known. So it's kind of interesting that there's sort of a dichotomy, is that they're useful for our work, but we know their receptivity to the organophosphates is a lot different.
So here's something that's been around, a weapon that's been around for 70 years or something like that.
Yeah.
And you're working on an antidote.
Right.
Have we really no antidote after all this time?
Well, that work is ongoing. So, yeah.
But But yes, we went a long time without an antidote.
Yeah.
And only now do we have the tools necessary to...
Well, the antidote, the first one was pralidoxine, and that's been out there for a long time, since the 1950s.
Oh.
Developed shortly after, and so yeah, that's kind of what's out there. Yeah, and that's in emergency medicine. The pralidoxine is what people have available as a treatment for.
But is it something like it's? You also mentioned the timing and the how. So the question is, if there is an antidote, why do we need more compounds?
Oh, well, that's a good question. Well, the main one is that what you saw in what we published, and the interesting thing is that the antidotes that are out there have a permanent positive charge and can't restore acetylcholine in the brain.
Oh.
Right.
I guess the diaphragm is the most immediate concern.
Right. Yeah. I mean,
but the brain is still
there. Right. And so the work that we've done and published, the focus has been on that aspect of it.
And the blood-brain barrier penetration issue is still one of the biggest hurdles for drugs.
Yeah. Well, generally, to get that, to have control over it, whether you want that or whether you don't want that.
That's right.
So the interesting thing with blood-brain barrier penetration is a separate issue, but in the way that progress in antihistamines has been made. So the first generation antihistamines, although the target's much different, it was the histamine, but then there's the muscarinic effects. So Benadryl, first generation antihistamine, it's centrally penetrating, obviously. You've experienced that. And then what changed in drug discovery was a realization that, the side effects were central. So what you saw in the second and third generation antihistamines was the placement of, all they had to do, well, it's short-changing all the work, but replace one of the functional groups with an acid. Right. So now you have a antihistamine with an acid, a non-drowsy antihistamine. So sometimes things like that, small functional group changes, can make all the difference in the world. The key that gets left out actually of the story of muscarinics. I know we're not talking about acetylcholinesterase anymore. Here's the main thing with muscarinics. There's a lot of muscarinics out there on the market. There have been for years. Atropine, scopolamine were early natural products, and now you just have a host for every application. People have been hoping for subtype selectivity. Mm-hmm. That's very hard to do. What the real work is in muscarinics is all in distribution. I mean, it's okay to seek an M3 versus M2, but what really matters is where that M2 is. Uh-huh. And so the difference in all the muscarinics is what is their distribution. So you have the smoking cessation. Something. Now we might have to edit this part out. But you have antimuscarinics that are ocular, and some that are not, obviously. And so, but the main thing, what matters for this work, what matters for a lot, is not just the medicinal properties in the lab, but obviously the compartmental distribution is everything when it comes to that class of drugs.
Jonathan. That's a good thought for us to end on. So thank you very much, Jonathan Bohmann and Jenny Hsieh.
Great, thank you.
This has been Neuroscientists Talk Shop.